Hi, and welcome to this run-through of the AQA A-Level Biology paper. That's paper 3 from 2019. Now, if you're doing this paper in 2024 as practice for your paper 3, this is a pretty good one to practice because it's recent, so the style of questions is pretty comparable to what you'll get in 2024, and also some of the content from this paper might be reused in 2024, whereas if you're doing a practice paper like 2022 or 2023, it's less likely, could still come up, but less likely to come up. So, let's get into this paper. Okay, first question. Describe how ultrafiltration occurs in a glomerulus. Nice, easy question to get you going. So I've put a picture over here just to give you a visual representation of that. So the first thing that we need to say is that ultrafiltration starts with the generation of high hydrostatic pressure. You don't really need to know exactly the mechanism of that, but there's a wider afferent arteriole here and a narrow efferent arteriole here. So basically more fluid is flowing into the glomerulus, which is a bundle of capillaries, than is going out. So you've got high physical pressure, so high hydrostatic pressure generated. And then that fluid is forced out of the capillaries through a few key things. It's forced through these little pores between the endothelial cells of the capillary. We can call them fenestrations. That means, like, I think it's from Latin, and it means little windows. Fenestrations. They're a special type of capillary with lots of fenestrations. Then it goes across this basement membrane, which in this diagram is labeled as connective tissue. It goes through the basement membrane. If you were doing OCR, you need to know a bit more detail about this, but basically it is just for your understanding, it's like a mesh of fibers, and it means that only small molecules can pass through this mesh. There's actually a molecular mass. Anything larger than 69,000 molecular mass can't get through. You don't need to know that, but what you do need to know is amino acids and sugars can pass through the membrane, but large proteins and cells cannot pass through this basement membrane. And then it goes past these podocytes, that are these blue things here, that basically hold that membrane in place. And you can actually sort of see that over here. This is a nice picture of the podocyte in blue wrapping around the capillary, holding that connective tissue, which is the basement membrane, in place. Okay, so that is what's going to be on the mark scheme, we hope. Let's look at the specific language. Make sure you hit all those keywords that AQA requires. So we've got, first of all, the high hydrostatic pressure. Could have said blood pressure, would have been fine. The fact that small substances pass out, you do have to name them. So water, glucose, ions, urea. Does it accept amino acids? Yeah, accept amino acids and small proteins, but not large proteins. Through gaps or fenestrations in the capillary. Mark scheme point 3. Oh, you have to say endothelium. You've got to say endothelium. That, I don't know if I would have got. In the capillary endothelium. If you're wondering, when do I use endothelium and when do I use epithelium? Capillary is always endothelium. Epithelium is more of an outer lining. So it would be alveolar epithelium and ciliated epithelium in the trachea. That's it. So endothelium, always capillary. And then through the basement membrane. Okay, let's move on. Okay, glucose and water are reabsorbed by the proximal convoluted tubule of a nephron. Put a tick in the box next to the correct ways in which glucose and water are reabsorbed. Okay, so let's break this down. Glucose by active transport and water against a water potential gradient. That's certainly not true. It is kind of active transport. It's kind of active transport / co-transport. That's why I'm reserving judgment on this bit first. Glucose by diffusion and water down a water potential gradient. No, I'm going to say no to that, and this is true. Glucose by facilitated diffusion. Okay, that's definitely no, because it can't go across the membrane on its own. And active transport and water against. Okay, it's this one. So this was by facilitated diffusion and active transport. That's the best answer. But against the water potential gradient, that's a big no. So it's this one. Okay, moving on. 1.3. The equation here shows the relationship between urine concentration in arbitrary units, y, and the mean length of the loop of Henle in millimeters. Okay, interesting. calculate the mean length of the loop of Henle in an organism that produces urine with a concentration of 16.56 arbitrary units. Okay, so the first thing to figure out is what is urine concentration in this equation. So urine concentration is Y, and it's 16.56. So we just need to basically solve this equation. So we're going to write 16.56 equals 0.72x plus 4. I'm going to solve this equation, so we're going to subtract 4. So that's going to be 12.56 equals 0.72x. We're going to divide both sides by 0.72, so cross that out. 12.56 divided by 17.4. Does it tell me how many SF? It doesn't, so I will probably give it a 17.4 millimeters. One mark. Let's hope I get that right. Yep, 17.4. On we go. Scientists investigated the relationship between the thickness of the kidney medulla of different species of mammals and the concentration of their urine. So you've got to have in your mind what the medulla is. The kidney is kidney-shaped, roughly like that. And we've got the kind of medulla is the inner region, and then we've got the cortex is the outer region. So that's the medulla of the kidney, and that's the cortex of the kidney. The glomerulus, glomeruli, plural, all the glomeruluses are in the cortex, and the medulla contains the loop of Henle. That's going to be important. So here's the results. The concentration of urine in different mammals, the thickness of medulla here, we can see clearly that as the thickness of the medulla increases, the concentration of urine, as measured in millimoles per dm cubed, also increases. Now, this question is a explain the pattern shown by the results question. So explain, not describe, but explain. That said, I might just say the trend just at the start. I mean, I've got enough space here, so I will probably start by saying the figure shows that as the thickness of the medulla increases, the concentration of urine also increases. Then I would say this is because. So why is this? Well, I wanted to drop in this picture over here to give you a little more in-depth explanation. Now, you don't need to know, you don't need to know the details of this for AQA, but this is the loop of Henle here, this whole thing like that, and then this over here is the collecting duct. Okay, and what I do want you to look at is just these numbers. This is the concentration in milliosmoles, which is just like a way of measuring the concentration of all solutes down here. So the concentration, not very salty, more salty, more salty, more salty, really, really salty, incredibly salty. The saltiness, and I'm talking about salt and other solutes, increases as we go down. And that's because the loop of Henle acts as it's called a hairpin countercurrent multiplier system. Basically, it makes things salty, and the longer the loop of Henle, the more salty it can make this space outside, down in the medulla. It's called the medullary interstitium. So we get more saltiness here. With more saltiness there, we have a very low water potential, and that means that water flows out of the collecting duct. Okay, so with a longer collecting duct, we can build up a greater concentration of salt in the space in the medulla, and that means we can have more water flowing out, a bigger water potential gradient. I suppose as well, just the fact that with a longer medulla, we're also going to have a longer collecting duct, so there's more length of collecting duct to reabsorb water. So those two things. Okay, so that's the explanation. Let's check the mark scheme for the exact language. So thicker medulla means a longer loop of Henle. And this increases the sodium ion concentration in the medulla. Therefore, so point 3, water potential gradient is maintained for longer. That's weird language. It's in brackets. So water potential gradient, I think you would say a higher water potential gradient would be accepted. It does actually accept that abbreviation. That's not normally allowed. So I would say a higher water potential gradient means that more water is reabsorbed from the collecting duct. So this is the key phrase, more water reabsorbed. But I think you do need to mention water potential gradient. I would think higher water potential gradient would be fine. Or if you didn't mention water potential, you could have just said more water is reabsorbed by osmosis. Down here. Water is reabsorbed by osmosis. Okay, let's keep going. Okay, describe the role of saprobionts in the nitrogen cycle. So saprobionts are kind of what AQA calls decomposers at A level. They are microbes and fungi, so bacteria and fungi, that do decomposition. Now we need to put some specific language in this. First of all, they do something called ammonification. So saprobionts do ammonification. This means, I'd elaborate, this means that they produce enzymes that break down and hydrolyze complex molecules like proteins and like DNA into smaller monomers and release ammonium. Okay, they basically digest dead stuff. So that should hopefully hit all the key words that we need. So they decompose, and you could have said hydrolyze. I would always say hydrolyze better, right? It's just a word that AQA loves, and you should like banish digest from your vocabulary and always say hydrolyze, or banish breakdown as well. Always say hydrolyze. So hydrolyze proteins, and they produce ammonia or ammonium compounds. You could say ammonification, that's what they do. Moving on. One environmental issue arising from the use of fertilizers is eutrophication. Eutrophication can cause water to become cloudy. You are given a sample of water from 3 different rivers. Describe how you would obtain a quantitative measurement of their cloudiness. So I definitely would want to use a colorimeter for this. I'm trying to think how I'd make sure to get 3 marks. I mean, you don't just say use a colorimeter, that's not going to cut it. But you'd put a sample of the water in a colorimeter cuvette, pass light through the cuvette, and record how much of the light is transmitted or absorbed. And then you would say, maybe I'd go on and say that cloudier water would give a higher value for absorbance or a lower value of transmission. You know, basically the more bacteria, because that's the bacteria and algae is causing the cloudiness, the more bacteria and algae that are present in the sample, the less light is going to be transmitted. I think that would be enough. If I had a bit of a space and I was wondering, is that going to be enough? I might say it may be necessary to dilute the river water sample first, potentially. I don't know. Let's have a look. Standardization. This is the bit which I missed. Okay, so colorimeter, yeah, that gives you 1 mark. Measure the absorbance or transmission of light, that gets you the 2nd mark. Some form of standardization. It kind of goes without saying, but you do need to say it. So, for example, you would use the same volume of water in each colorimeter cuvette. make sure that you're zeroing the colorimeter every time, or make sure that you shake the sample, or use the same wavelength of light every time. So just some form of control variable. Possibly worth kind of remembering these as general control variables for colorimetry, because colorimetry can come up in other ways. Got it. Okay, great. Okay, question 3. Here is a photograph of a heart, a dissected heart. We've got valve A, chamber B, and we've got left atrium. So I presume that probably means, if that's the left atrium, this is probably the left ventricle. Okay, that's that. And then valve A, I would say, is the atrioventricular valve. Atrioventricular. That's the easiest valve to remember. Well, it's easier to remember that than remembering bicuspid and tricuspid. Actually, this would be the bicuspid valve, is on the left side. The atrioventricular is kind of easier to remember because it applies to left and right, so you don't have to remember as many things. So I would say that. Chamber B, left ventricle. Double check. Dum, dum, dum. Yep. Atrioventricular, left ventricle. You could say bicuspid, but it's probably not worth trying to remember that, unless you are planning to do medicine. In which case, you'll have to learn it sooner or later anyway. Okay, safety precautions that should be followed when dissecting a heart. Okay, so when dissecting a heart, we're going to be using scalpels. So think about safety precautions to do with scalpels. When you're cutting, you want to use a sharp scalpel. That actually reduces the chance of injury. And you want to cut away from your hand. You want to wear gloves. You want to wash your hands after the procedure. And you also want to dispose of the heart safely in a biohazard bag, for example. That's kind of like a after-the-fact safety precaution. So that should be enough. You only get 1 mark for the 2 safety precautions. Feels ungenerous. Let's see. Yeah, you've got to have 2 of these. Sharp scalpel, wash hands or wear gloves, disinfect the bench or equipment. This is cover any cuts, that is sensible. Cut away from self or others or on a hard surface and dispose safely. Interestingly, it says ignore goggles, because I imagine some people might have said that, but there's no dangerous liquids involved, so that's why goggles is not a prerequisite. Yeah. And then this is not specific enough. Take care. How do you take care? So, for example, if you are taking care with scalpels, you need to actually say how to do that, which is this: cut away from others, cut away from yourself, not towards yourself. You need to describe in more detail. Explain how valve A in figure 2 maintains a unidirectional flow of blood. This is about like how it closes, basically. So valve A. So I would say as the ventricle begins contraction, the pressure in the ventricle rises, and I would say it then becomes higher than the pressure in the left atrium, and this then causes valve A to close, so that blood cannot flow back into the atrium, and then I would say instead the blood exits via the aorta. Could put through the semilunar valves out into the aorta. Let's look at the mark scheme for that one. I only get the 1 mark because I need to talk about the first bit. So initially, I probably should have said this. So initially, pressure in the atrium is higher than in the ventricle, and the valve is open, meaning blood flows from the atrium into the ventricle. And then, when contraction occurs, the pressure in the ventricle is higher than the atrium, causing the valve to close. So you need to talk about when it's open and when it's closed. You could also talk about the tendons or tendinous cords preventing the valve from turning inside out. Actually, a student asked me a question about this the other day, so let me just show you on this diagram right here. Let me get rid of my doodlings. You can see all these little tendons here. If I pinch in a little bit, that basically prevent the valve from inverting. All these little things here, they're just holding the valve in place so that the valve, when it can kind of slam shuts, it kind of goes like this, boom, like that, but it can't flip round and invert. Okay, so it's held, it can't go further than that. So it closes, but the valve stop it from like doing that. Yeah. Let's move on. Okay, now we're looking at some data, a study. So whenever we look at some data or a study, we want to take time to read it and sort of start to think about areas of the study that were good or like things that could be improved, because we might get asked about it later. And we might be asked to evaluate things. A research scientist investigated the effect of caffeine, so that's a stimulant, on heart rate in human volunteers. The scientist divided volunteers into 3 groups. Each group was given the same volume of fluid. Okay, so that's a control variable. Each member of group I was given a sports drink containing, I'm going to start color coding, sports drink containing caffeine and sugar. That is group I, and that's this one. So I'm just going to go over this because I really like colorful things and I can't really process information if it's in black and white. Maybe you're the same, who knows. And then we've got caffeine and no sugar. Let's do blue for that one. Caffeine and no sugar. And that is group J. Okay. And then what's the other one? Water, is it? Yeah, water. So that's like a control, basically. I'll just do that in yellow. And not much happens with them. Okay. The scientists recorded the volunteer's heart rate before the drink was given, and for 120 minutes after the drink was given. Okay, so the reason they did it before was to work out the average resting heart rate, possibly. Before. Now, it doesn't actually say resting heart rate. We don't actually know. Okay, so that's already something thinking that's maybe something that could be improved. So were the volunteers doing something standard? Were they sitting down or were they jumping up and down? That would obviously affect heart rate. It seems strange. I'm also thinking it seems weird that the heart rate goes down 15 minutes after drinking. I guess maybe that's like the digestion. More blood flow diverted to the stomach. That wouldn't cause heart rate to go down. I'm intrigued by that. I don't know why it is. Let's move on and maybe we'll get asked about it, maybe we won't. Okay, 3.4. Caffeine affects the autonomic nervous system. Suggest how caffeine could account for the results of group I at 60 minutes. Okay. So, group I, up here, at 60 minutes, this is an increase in heart rate. So this is the green one. 60 minutes, we've got a heart rate of 7. So the heart rate has increased by 7 beats per minute. So we want to explain this. The question actually doesn't ask us to describe, just explain or give a reason for that. So I think the clue is an autonomic nervous system. The question obviously wants us to talk about it. So what is the autonomic nervous system? Well, we've got 2 different branches. We've got the sympathetic and the parasympathetic branches. So you need to remember which is the speeding up and which is the slowing down. Sympathetic speeds up heart rate, parasympathetic slows it down. So we want to talk about the sympathetic nervous system. So we say that, I would probably say that caffeine increases the frequency of action potentials traveling from the cardiac center down the sympathetic nervous system to the sinoatrial node. Okay? Hopefully that's going to do it. Let's see. Okay, so more action potentials. More action potentials. I'll probably say action potentials rather than impulses. Okay, electrical impulses is slightly better, but more action potentials along the sympathetic nervous system to the SAN, thus increasing the heart rate. Yeah, you have to have the idea of more frequent action potentials. Okay, let's keep going. Before taking the drink, the mean heart rate of group J was 68 beats per minute, okay? 15 minutes after taking the drink, the mean volume of blood leaving the hearts of group J was 4,700 cm per minute. Calculate the mean volume of blood leaving the heart at each beat 15 minutes after taking the drink. First of all, this question is kind of about cardiac output. You do get asked questions about this. Cardiac output, it doesn't actually say the words cardiac output, but cardiac output is equal to the stroke volume times the heart rate. And we've been given the cardiac output. We've been given 4,700. And we've actually been given, we're trying to figure out what the stroke volume is, the mean volume of blood leaving the heart at each beat. But we also need to figure out what the heart rate is first. So we figure out what the heart rate is, and then do 4,700 divided by the heart rate, and that's going to give us our stroke volume. So let's figure out what the heart rate is. Well, we know that it starts at 68, and 15 minutes is right here. And it's, which group was it again? Was it group J? Group J, which is the blue one. I forgot to highlight all these other ones. That's the blue one. Group J. So, Group J, blue one, wow, it's gone down quite a lot, hasn't it? So that's down to -4. That's 1, 2, 3, 4. So that's -4 beats per minute. So if we started out at 68, -4 is going to give us 64 BPM. So therefore we do 4,700 divided by 64, and that's going to give us our number, which is 73.4. I took too long on that. That was one mark. 73.4. Yep, 73.4. Okay, the increase seen in group I could be due to the combination of caffeine and sugar. Suggest one drink to be given to an additional group that should be investigated to find out if this is true. Just caffeine, I think. Yeah. So let's look at the top here. We've got caffeine. Oh no, just sugar. We need just sugar. So we've got caffeine and sugar as one combination. We've got just caffeine, and then we've got water. What about just sugar? We haven't got just sugar. So is the big boost that's seen in green, is that because of the combination of caffeine and sugar, or is it just because of the sugar? It's difficult to tell. So that's the answer, but the mark is going to be for the explanation. So I would say the group should be given sugar, and I would say the reason is we would want to see the effect of sugar in isolation without caffeine. And I might say the one group shows us the effect of caffeine on its own, but we don't know the effect of sugar on its own. And then we definitely want to have, say, for comparison. They love that language, for comparison. So sugar solution only, yeah, or a drink with sugar and no caffeine. To prove that sugar alone is not causing the increase in HR, or to see the effect of sugar on its own, I think. Yeah. Okay, question 4. Mitochondrial DNA, mtDNA, is a small circular DNA molecule located in a mitochondria. It is that many nucleotides long and contains 37 genes and a control region. 37. I thought it was more. Sorry, less. Okay, 37 genes. Sports scientists investigated whether a mutation in the control region in human males was related to an ability to exercise for longer. Okay, the males in group C had thymine, group T had thymine, T for thymine, and group C for cytosine. That's nice that they've made that easy for us to remember. T for thymine, C for cytosine. Okay, the control regions of group T and C were the same length. So this is just a swapping or a substitution mutation. That's the answer to the first question. What type of mutation is this? It is a substitution, which kind of means a swap. Group T and C completed the same 8-week training program. The following measurements were taken at the start of the 8-week program and again at the end. Okay, VO2 max, measure of maximal oxygen uptake. So that is going to be to do with lungs, I suppose, mainly. I'm not a sports scientist. Lungs, capillary density in the lungs, all sorts of things. And then citrate synthase activity. And this is an enzyme that is in the Krebs cycle. It catalyzes one of the steps of the Krebs cycle, I think. The scientists then calculated the percentage increase in each measurement in both groups. Also, Krebs cycle, that is obviously involved in aerobic respiration. Okay, needs oxygen for the Krebs cycle to run. Scientists then calculated the percentage increase in each measurement, so it's percentage increase in both groups. Okay, so group T, 8-week training program, the same 8-week program. Okay, cool. Okay, so basically VO2 max has gone up more and citrate synthase has gone up more. Something like, I would say, 18% is that one, and this one is going to be, I think, 13, but I would probably get my ruler out if I was doing this in a real exam and really try and accurately identify that, but I think that is right, 18 and 13. Over here, I think that is 68, and that's bang on 40, I believe. Okay, what is the actual question they ask? Okay, down here. 4.2. A student concluded from both figures, 4 and 5, that training has a positive effect on VO2 max and citrate synthase activity. This looks to me to be more points for than against. Evaluate the student's conclusion. I reckon there's probably going to be maybe 2 marks for and one or 2 marks against. I'll probably say 2 things for and 2 things against, just to cover myself, just to make sure. So let's try and get a plus, a negative, and a negative. All right, let's find a plus first. Okay, so first of all, we can say that both group T and C showed an increase in their VO2 max after training. Maybe you just give the data, 18% increase and a 13% increase. So training does seem to increase VO2 max. However, as a negative, we could probably say the same thing about citrate synthase. So increase, the data increases basically. I wouldn't say it like that, but that's the key first point. Maybe we don't have no idea of standard deviation. No statistical test has been done. And then I'd probably say data increases VO2 max and citrate synthase activity. Probably say that. So, another thing we don't really know. Oh, it is all men, isn't it? It's all men. Human males. So, but we don't know much about the age of the men. We don't know if there was a controlled study in terms of the age of the men in the different groups, we could say, or the study was only done in men. Yeah, that might be a negative as well. Study only done in men, not in women, because this student says that generally causes a positive effect. Right, let's see the mark scheme. Okay, so the first mark for the data, that is good. Got that one. Both VO2 max and citrate synthase activity increase for both groups. So no statistical test. We've got to make sure that when we give this point, we kind of elaborate and use the right language. No statistical test, so we don't know if the differences are significant. And then might not be true in females. Said that. Okay, I was thinking about this. Maybe this only 8 weeks of training, so we don't know if it's a long-lived effect. It feels like quite a lot of training, but it's not a year, so we don't know if it's going to keep going. Okay. Let's just ignore standard deviation. Okay, let's keep going. .3. The mitochondrial DNA, mtDNA, control region is an area of mitochondrial DNA that is non-coding. This region stimulates the synthesis of both mitochondrial DNA and mitochondrial messenger RNA. Okay, so it basically controls replication and, sorry, handwriting's terrible today, and transcription. Use this information to suggest 2 reasons, maybe it's one about each thing, why mutation at nucleotide position 1616519 could lead to the differences seen. 2 regions. Okay, so it could affect the frequency at which, well, it could affect the frequency at which RNA polymerase enzymes bind and initiate transcription. So it could affect the level of expression of those, however many genes it was, 37 genes found in the mitochondrial DNA. I suppose it could also, no error. I suppose it could also affect the frequency of mitochondrial DNA replication, such that maybe the mitochondria could potentially have 2 or even 3 copies of its mitochondrial DNA within one mitochondria, which also could affect the level of the proteins produced by those genes. Can I think of any other ones? That's about it, I think. Let's see what the mark scheme says. Okay. So, yeah, it is about those 2 different things that kind of guided us towards. Less transcription or more transcription of the mitochondrial genes, and therefore less, you could have then linked it and said less translation. It's the same mark point. Mark point 1, less mitochondrial replication or production. I wonder if it would have accepted less DNA replication or more DNA replication. I don't know. Not sure whether that would have been accepted or not. I think perhaps, but I'm not sure. Okay, moving on. The sports scientists investigated whether there was a correlation between the percentage change in VO2 max and the percentage change in citrate synthase activity. doesn't, I mean, maybe a little bit. Maybe there's a line that goes a bit like that. Slightly up, but it's pretty vague. Okay. Having thymine nucleotide position 16519 in group T causes an increase in ability to exercise for longer. Evaluate this conclusion. Right. Use all the data. This is a lovely evaluate one, isn't it? Okay, so first, causes. If you've got that word causes, no statistical test is definitely going to be on the mark scheme. I'm also thinking that we actually haven't measured what they're actually talking about. We haven't measured, I suppose, we've measured VO2 max and citrate synthase activity, but we actually haven't measured what this person is talking about, an ability to exercise for longer. We haven't quantified that. And group T, can group T exercise for longer? So for group T, so this is where we need to use this data here. So group T does have a larger increase in VO2 max and a larger increase in citrate synthase activity after training. So that's probably going to be worth a mark. That's probably worth a mark. So talking about group T, higher increase in VO2 max plus citrate synthase activity. Also, if we want to bring in this graph here, this is correlation, not causation. So there is some correlation between the VO2 max and the citrate synthase activity, but there's been, so correlation, not causation. For figure 6, and that's CS. Does that lead to VO2 max? Who knows? We could mention the fact that we would do a Spearman's rank test. I think that's probably going to get it, I think. Let's see. Let's see the mark scheme. Got 4 things there, 3 points. Hopefully that's enough. Okay. So from figure 5, that's the first one. Group T have increased CS activity for the Krebs cycle, and actually you can get 2 marks from this. From what was figure 5? I need to go back. Figure 5. Were there 3 figures? Figure 6. Sorry, figure. That's figure 4 and 5. That's the same. Okay, both those ones. Okay, 4 and 5. Okay, so you get a mark for talking about figure 4, and a mark for talking about figure 5. It does look like you have to explain it. Yeah, so increased CS activity for the Krebs cycle, increased VO2 max, so more oxygen available, or so more aerobic respiration, or delayed anaerobic respiration. Okay, so 5, this is what I was talking about for 5. Accept the idea that they did not measure the ability to exercise for longer. We didn't actually record that. We just recorded this and this. There was no actual quantifying measure of the time until exhaustion. What else? What else did I miss? Okay, limited correlation. I said there was some correlation. Maybe I should have been more critical. No correlation. No correlation on figure 6. There might be other differences that cause. There could be other differences in the control region of group T that cause the increase. That's interesting. So it might not just be thymine that has mutated. There might be other mutations that we weren't looking at. So how many marks would I have got on this? I think I would have got this and this. I don't know if I would have. If I'd written it down, I probably would have said this. I would have got 5. I think I would have got 3. Yeah, I would have got 3, just. Okay, let's keep going. Tough question, that one. Especially this bit. That's my takeaway. That is not good enough for a correlation. Yeah, it looks pretty much 0, maybe 0.1 correlation or something. That's a Spearman's rank value, by the way. It goes between 0 and 1. 1 is perfect correlation, 0 is none, so it would be pretty low. Okay, the crown of thorns starfish. Ooh, I like these. Here's one. Is one of the main causes of the decline of the world's coral reefs. Marine biologists used a choice chamber, interesting, to investigate the effect of flashing and constant light on the behavior of the crown of thorns starfish. Shows the results. Table 1 shows the results as they presented them. The p-values show results from a statistical test. All right. Okay, so the crown of thorns were either moving towards the flashing light or away from the flashing light. There was a slight increase in more of them were moving away. The statistical test they did gave a p-value of 0.69. High p-value means not significant, so this is not significant. Whereas if the light was constant, 12 moving towards 38 moving away, that is quite a big difference, and that gave a very highly significant p-value, 0.02. That is significant. So probably, I'm guessing they used a chi-squared test here, because like they would have just compared the observed minus the expected. So the null hypothesis would be that there is the crown of thorns, COTS, show no taxis response towards the light. Either flashing or constant. So there would be no difference in their movement. There would be no difference in their movement towards or away towards the light. I won't put that in brackets. I.e., there would be, i.e. they would not move. There'd be the same amount moving towards and away. Yeah, okay. So this would, I think this would get the mark. There is no difference in behavior or movement with constant or flashing light. Okay. The reason I'm pausing there is because I feel like you would have to have 2 separate null hypotheses for this investigation. You'd first of all have a null hypothesis about flashing lights, and then you'd have a null hypothesis about constant light. That's why you've got a p-value. Your p-value will either like make you accept your null hypothesis or disprove it. And so I think there's like, actually there'd be a null hypothesis for this set of results and a null hypothesis for this set of results. And it looks like we accept the null hypothesis for this one, reject the null hypothesis for this one, because this is significant. That's why I was kind of slightly confused by why they had this sort of general type of light. This, I think, is more accurate. Okay, let's keep going. The natural habitat of the crown of thorn starfish is coral reefs or tropical oceans. So he has 2 factors. Constant 2 factors that should be kept constant in the choice chambers so that crown of thorn starfish display normal behavior. Okay, so tropical oceans is what we need to think about. So temperature, And I'd probably say salinity. Salinity of the water. That's probably what I would guess. pH of the water possibly could be on there. Okay, salinity and temperature, yeah. Amount of food, pH of the water, oxygen concentration or carbon dioxide concentration of the water, intensity or the wavelength of the light. Okay, let's keep going. 5.3. A journalist studying Table 1 suggested that either type of light could be used to cause crown of thorns starfish to move away from the coral reefs. Evaluate the journalist's suggestion. Well, this is silly. So this either type of light, no. No, certainly not. So what we need to pick up on is what type of light would be effective and what wouldn't. Constant light would be effective. Flashing light would not be effective because it is an insignificant result because the p-value is high, higher than 0.05, so that is larger than 0.05, and this is smaller than 0.05. So I think there's going to be marks for talking about this, for saying it's bigger than the critical value of 0.05, and in this case it's smaller than, so this difference is not significant, this difference is significant. That's probably going to be 2 marks. Other things, this seems a bit silly. We haven't done this in like we've only done this in a choice chamber, and we don't know anything about the light intensity, I would say. This is kind of my like practical consideration. I don't know if it'd be in the mark scheme. Basically that this is like a small choice chamber where you've got a flashing light that makes a constant light that makes the starfish move away. But if you were doing it in an ocean, would that constant light be bright enough to travel any distance? Like, because a coral reef is a big thing. How many lights are you going to have? Seems a bit of a silly, like a very silly suggestion, to be honest. So that's probably what I would go for my third point. And do we know, are there different species of crown of thorns starfish that might be affected in different ways? And would it work in the daylight or not? I'm not sure if this is only done at nighttime. Would it work in the day? I don't know. Let's have a look. Gosh, okay. It does give you this mark for yes, the movement is away from either. But the significant movement is only away from constant light. And the movement away from flashing light is not significant. Any other marks on that? No. Okay, so you do, strange, I didn't think that you would get any marks for actually saying the fact that both of them do move away, both of them do cause some movement away. Negative phototaxis to both types. The movement away from constant light is significant. You have to say why, because the p-value is lower than that. And movement away from the flashing light is not significant as the p-value is higher than that. Okay. One of the reasons COTS can destroy coral reefs in a shorter time is because COTS move quickly, allowing them to move from one reef to another. Table 2 shows the maximum speeds recorded of COTS in constant light. So this is when it's moving away, showing negative phototaxis. COTS moving towards the constant light. Maximum speed 259. COTS moving away from the constant light, maximum speed 564. Okay. Calculate the shortest time one COTS would take to move up a coral reef from 66 meters underwater to 18 meters underwater in hours of daylight. Why is that relevant? So, okay, well, 66 to 18. What's that going to be? Get my calculator out. No capacity for mental mass today. 48. 48 meters difference. The maximum speed okay. Oh, I see. Okay, so these two values are away from constant light or towards constant light. In hours of daylight, the light in a coral reef comes from above. I think this is what this is talking about. So, like, you know, the light is coming from above. Ta-da! So they're moving towards the light in hours of daylight. So we want to use this value, 259. So we need to take this 48 meters is actually 48, how many millimeters in a meter? 1,000. Yeah, 48,000. So this is 48,000 millimeters. And we need to divide that by 259, which gives us our time in minutes. 48,000 divided by 259, which is a lot. Which is a lot of minutes. 185.3 minutes. Then we divide that by 60 to get our time in hours, and it's 3.09 hours. Oh, is that right? Is that right? Ah, gosh, I almost lost the mark there. I just missed that. Bad exam technique for me. Give your answer to the nearest hour. So, to the nearest hour, rub that out. Okay, yeah, so correct answer of 3 hours is 2 marks. And if you had forgotten, well, I almost forgot there, that would have only been 1 mark if you give them 3.09. If you'd used 564 instead of that's like moving away from constant light, then you could have got 1 mark for that. Question 6. Uncontrolled cell division can cause tumours to form. Figure 7 shows the growth pattern followed by a rare type of tumour. Okay, here we have time in years along the x-axis, and then y-axis is log 10 number of cells in tumour. So that means, in case you're unsure, like over here 2, this is 10 to the 2. This is 10 to the 1 cells, 10 to the 3, and so on. So this would be 1,000,000 cells right here, 10 to the 6. Use figure 7 to calculate the percentage of maximum growth this type of tumour reaches before it can be detected. The percentage of maximum growth. Okay, so before it can be detected. Okay, so there's 2 things. When can it be detected, and what is the maximum growth? So it can be detected at this point. So that's 10 to the 9 cells. And then the maximum growth is up here, which is 10 to the 12 cells. Okay, so that means the percentage is simply 10 to the 9 divided by 10 to the 12. So if we do that on our calculator, 10 to the 9, I can do this sort of longhand actually, can I? Yeah, so that's 1 billion, 10 to the 9. And then this is going to have 12 zeros, so it's going to have 3 more zeros basically. Boom, boom, boom. We cross out all those zeros, we cross out all those zeros, so it's 1 divided by 1,000, and then times 100 to make it a percent, times 100. I'm going to do all this longhand, so it's 100 divided by 1,000. So cross two zeros, cross the 1 over 10 percent, so 0.1%. Did I correctly do that longhand without any calculator? Let's see. Is it 0.1%? Is it 0.1%? It is 0.1%. You should have probably done that with a calculator. I don't know why I decided to just work that out, but yeah, 0.1%. So, moving on to 6.2. Figure 7 can also be used to calculate the age of this type of tumour. At diagnosis, a patient had 3.98 times 10 to the 11 cells. 3.98 times 10 to the 11 cells. Okay, so what we're going to need to do. Okay, so we do 3.98 times 10 to the 11. Work that number out, whatever that is. Well, it's just 3.98 with lots of zeros after it. 3.98, big number, and then I want to do log 10 of that. So log to the base 10 of this long number is going to give us the exact power of 10. So 3, 9, 8, 1, 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. And then I want to do log 10 of that. Boop. And it gives me 11.599. Okay, equals 11.6. Now I go to this table up here. Sorry, to this graph. 11.6. That's 11.2, 4, 6, 8, 10. So it's there. 11.6 is right there. And then I go down all the way down to here. So it's 4.8 years. 4.8 years. Does it say to the nearest year? 4.8 years. Is it going to be right? Let's hope so. Yes. Somewhere between 4.7 to 4.9. So yep, 4.8 is correct. Phew. All right, keep going. Trexol is a drug that can be used to slow the development of various forms of cancer. So slow development, so maybe not necessarily kill, but slow. Trexol slows its cell division by interacting with an enzyme called dihydrofolate reductase. I've heard of this somewhere, I can't remember why. DR is involved in making nucleotides. The substrate for DR is folic acid. Okay, so Trexol and folic acid. They have a very similar shape. Interacting with an enzyme. And it looks to be, because they're such a similar shape, is most likely going to be competitive inhibition. So this is Trexol, and this is folic acid. Use the information provided to suggest how Trexol slows cell division. Okay, so there's an enzyme. Let's just draw it over here that has an active site that kind of fits this molecule. Here's my enzyme. There's my enzyme. Now, either folic acid can get into that active site or Trexol can get into the active site. So you need to say the two substrates have a very similar shape and would both be complementary to the active site of the enzyme. So Trexol acts as a competitive inhibitor. So if Trexol binds to the active site, it means that the substrate folic acid cannot enter the active site, so no enzyme substrate complexes can be formed, and the rate of reaction will be slower. Smart scheme. A competitive inhibitor is what it is. So it is able to fit and bind at the active site, so less folic acid or substrate attaches, or fewer enzyme substrate complexes. I've forgot to add that last bit of the question. So the question, let me just go back to the question for a second, and I'll come back to the mark scheme. The question is, use the information provided to suggest how Trexel slows cell division. I didn't see that, I was going too quick. Not just how it inhibits the enzyme, but how it slows cell division. So it inhibits the enzyme, and that means that fewer nucleotides are made. So therefore, there are not enough free nucleotides for semi-conservative replication. Question 6.3. Well, no, we still more data. Doctors investigated how the concentration of Trexel given to patients affected the growth of lung tumours. The doctors measured the volume of tumours at the beginning of the study and after 8 months. Okay, so a change over an 8-month period. The bars represent plus or minus 2 standard deviations, and this value of plus or minus 2 standard deviations from the mean includes over 95% of the data. So if the standard deviation bars overlap, there's not a significant change, and if they don't overlap, then there is a significant change or a significant difference. The scientists measured the percentage change in tumor volume. Suggests why they recorded both percentage change and tumor volume. Well, percentage change so that we can compare the effect of Trexel on tumors with different initial sizes. It makes the results comparable. Tumor volume, so we can measure the absolute size of the tumor. Gosh, I'm not quite sure. Why do we measure tumor volume? What do we say? Just because? Why not? Well, because you can't calculate percentage without the tumor volume. How can you calculate the percentage change without calculating the tumor volume? In order to calculate the percentage change, it would be impossible to calculate percentage change without the tumor volume. Is that the answer? If it is, it's a strange question. Percentage change to allow comparison, as tumors may differ in volume or size. Oh, why volume as opposed to shape? Okay. I get it. So some tumors, I didn't understand what it was getting at. So some tumors might be this kind of shape. So if you were to measure the length of that tumor that way versus that way, how are you going to decide which way to measure it? It's best to kind of measure the total volume, because it is a good indicator of the number of cells in tumor, because each cell has a roughly similar volume. Okay, that is an interesting question. I haven't seen that before. I like it though. So volume is a much better indicator of the number of cells rather than length width, because tumors can be irregular in shape. Nice. (6.5) A lung cancer patient received 15 milligrams of Trexel per week. After treatment, the diameter of his lung was 35.8 millimeters. This is after the 8-month treatment. Assuming the tumor was spherical, use the mean percentage change in tumor volume shown in figure 10 to calculate the volume of the patient's tumor before treatment with Trexel. Oh, this is long-winded. Okay, so D equals 35.8, therefore R equals, what is that? Divided by 2. Can't do that in my head. 35.8 divided by 2 equals 17.9. So the radius is 17.9. So the volume equals 4/3 pi r cubed of that. So 17.9, that seems like a lot. 24,024 millimeters cubed. but that's after treatment. So we want to know the volume before, and this is, okay, so 15 milligrams of Trexel. So let's go back up here, 15 milligrams of Trexel. So this, if we zoom in, what kind of reduction in tumor volume can we get? It looks to be bang on minus 25%. So minus 25% is the change. So this actually here, this equals 0.75 of the starting value, or 75% of the starting value. So if we, because it's reduced by 25%, so we need to divide that by 0.75 to get the answer. Divide by 0.75 equals, I've got 32, does it tell me how many decimal places or significant figures? No, 32,032 millimeters cubed. Oh, I used pi for my calculator. I didn't use 3.14. I hope that's not going to get me a wrong answer. 32,000. What did I say? I think that's right. 32,000. 032. 32,032. Yep, so that would be in that range. Okay, cool. Great. (6.6) To reduce the size of tumors, would it be better to use 30 milligrams of Trexel per week or 20 milligrams of Trexel per week? Well, my initial thoughts is use 30, but let's go back and look at the data. I think it is better. Okay, so 30 is better. So I think we need to talk about the error bars, I think, here. So 20, we're in this range. 30, we're in this range. There is quite a high standard deviation, or a large error for 30, but it is significantly lower than 20. If we look at the positions where those error bars kind of end, that's where that one ends, and that's where that one ends, so they don't overlap. So 30 does produce a significantly greater decrease in concentration, decrease in volume, and it's minus 25% versus, what's that? It looks like minus, is that minus 75%? Yeah, so it's a 75% reduction in tumor volume versus a 25% reduction in tumor volume. There is a bit of an error associated with this, but it is a significant difference. Oops. I might also say that, is it an evaluate? No, it just says explain your answer. So the number of cells in the tumor would be reduced. I think that's it. I think probably the marks are for the kind of error bars thing, I think. Yeah, there is a significantly greater reduction. Error bars do not overlap. That's an interesting point. So in some cases, there was a 100% reduction in size of tumors. That's interesting. This is a question. I don't like this question because the command word of the question is not the right command word. AQA, come on now. This is an evaluate question. This should be, someone says it's better to use 30 milligrams of Trexel per week versus 20. Evaluate this statement. Because actually the mark scheme has marks for and against, which is characteristic of an evaluate question. So I feel like this is AQA not really writing their question very well. Against, we don't know if there's side effects. We don't know if there's an extra cost associated with, well, there will be an extra cost because we're paying for more of the chemical. And there is a lot of variation. I really feel like this question should have been an evaluate and not an explain. The command word is all wrong. AQA not helpful for me or for students. So I guess just be aware sometimes that they might not really tell you it's an evaluate question, even though it is. Annoying. Okay. Trexel can also be used to slow the development of rheumatoid arthritis, kind of like what, generally speaking, more elderly people get in their fingers and stuff like that. Scientists investigated the effectiveness of Trexel as a pain relief treatment in 12 rheumatoid arthritis patients. I feel like that's a small number. All of the patients were female. I feel like that's maybe not great for a study. Randomly divided the patients into 2 groups. So there's only 6 people in each group. And what do they get? Trexel, Group R received Trexel tablets for 35 days. Group S was a control group. Did they get any tablets? We don't know. They asked the groups to rate their pain on a scale of 0 to 10. Okay, so this is self-reported data. So that's not very, not ideal. At the start, and then every 7 days for 35 days. So 35 days is 5 weeks. Yeah, 5 weeks. And they calculated mean scores for each group. Okay, so Group R reports their pain as going down to 5.1. A little bit of fluctuation, but basically going down. Group S also reports it largely going down. Apart from age and general health, give 2 important factors when choosing patients for this investigation. We've ruled out a lot of stuff that we would normally talk about. So we can't talk about age, can't talk about health, general health, and we can't talk about whether they're male or female, because that's already up there. We can't, God, I don't like this at all. I mean, is it about their arthritis? We know they've got arthritis. Could it be how bad the arthritis is? How much, how bad their arthritis pain is to start with? Maybe. What else? We've got, when we're looking at patient studies, we're thinking about age, sex, ethnicity. Ethnicity is one. So let's do that. Okay, so type of degree of arthritis and ethnicity. What else? Are there different types of arthritis? Maybe arthritis of the hands, arthritis of the knees, body part affected by arthritis, maybe? What we got? Medication. Yeah, okay. So severity, duration of arthritis, type of arthritis. Weight we could have had. Ethnicity was there. Weight or body mass, that's an important one. Missed that one, that should have been obvious. And any other medications that may interact. Medications, body mass. (6.8). Here we go, here's an evaluate. The student analysed table 3 and concluded that Trexel was effective in reducing pain in arthritis patients. Was effective in reducing pain. Okay, well, so evaluate here, I feel like this is going to be let's always try to, if it's a 3 mark, we still try and say 2 positives and 2 negatives, because, we say 4 things, even 1 is wrong, we get 3 marks. So let's go for the 2 positives first. The 1 positive, there was a large reduction in the group R from 9.7 to 5.1, and the overall trend was decreasing. That's a positive. Can I even think of a second positive? I can think of maybe 3 negatives. A negative. The control group also reported a decrease in pain. Another negative. We don't know if the reduction in pain of the group R was statistically greater or statistically significant when compared with the reduction in pain of group S. Is it greater than a placebo? Another thing, group S, did they even receive a placebo pill? They probably should have done in the study. If they just had a control group and they didn't get any placebo pill, then It's not an ideal study. It would be better if it was a placebo. Third thing, there's only 12 patients. Fourth thing, only female patients were used. And that's 4 things. That's probably enough. Oh, and a fifth thing, it was only done for 35 days. So we don't know if it would continue on beyond 35 days. That's plenty. So 4. There is only one 4. Okay, we're struggling there. Pain decreases more compared with the control group. Against small sample size. The mean score of pain in control group is also lower, or it also reduces. There's no statistical test. Are there more points down here? Yeah, I did mention this when we were talking about the question, but I forgot it when I went to the answer. Pain is a subjective measurement, so the self-reported pain as a measurement is a tricky one because we can't be 100% sure that it's objective. That's why pain studies are quite difficult to do because it's difficult to quantify it. It is in someone's brain, after all. Okay, question 7. This is section B, and it's the essay. You've got 45 minutes to write an essay. The first thing to do is to try and pick what essay is going to be the best for you. So just quickly have a think about the two. We've got the importance of DNA as an information-carrying molecule and its use in gene technologies. Okay, there's lots of things that spring to mind about that. And then we've got the importance of bonds and bonding in organisms. First thing to do, I think, is just try and think of 4 separate topic areas that we could talk about. So we could talk about DNA structure and replication. That's one topic area. We could talk about transcription and translation, and the sort of flow of information from the DNA into the protein. And then gene technologies, we could maybe talk about genetic engineering, and we could talk about electrophoresis, and screening. Down here, the importance of bonds and bonding in organisms, I would find this maybe a little bit harder. Bonds and bonding in organisms. Goodness. Okay, so bonds, I'm thinking about biological molecules. So I'm thinking about carbohydrates and proteins and polymerization. I'm maybe also thinking about water, hydrogen bonding, bonds and bonding. What else? We'll talk about temporary bonds, like bonds between a hormone and a receptor protein. Do we count that as bonding? I think I'm already pretty much convinced that I'm going to do the first essay because I'm struggling here. Bonds and bonding. I mean, there's so many things. The thing that's difficult, and I'll now add in a picture here. The thing that's difficult is that you need to make sure that when you're picking 4 different topic areas, they are about different topics. And I find this a bit frustrating because it does actually mean that you need to know which bits of content are in different blocks or different subtopics, which I really feel like you shouldn't really be spending your time doing, but you have to because it's important for your getting as many marks as possible in the essay. I just feel like it's a bit of like it's a technical thing that I would rather you be concentrating on amazing things about biology and less this, but you need to do this. So what I was thinking about there for gene expression is if we talked about 3.8.4 recombinant DNA technology, is it a different topic to genetic fingerprinting? It actually is, because it has these sub-spec points, 3.8.4.1, 3.8.4.2, 3.8.4.3. Basically, that means, and you can just remember this, that all of 3.8 gene expression, all of those different spec points within that, are different topic areas. An example of something that isn't a different topic area would be if you talked about, I don't know, if you tried to talk about two different aspects of photosynthesis, the light-dependent reaction and then the light-independent reaction, they would be under that same topic area. So actually that does work in terms of topic selection. This would work. So for the DNA essay, I would pick something like recombinant DNA technology, so genetic engineering, genetic fingerprinting, and then I'd also pick nucleic acid sort of DNA structure and replication, and then over here I'd pick DNA and protein synthesis, so basically transcription and translation. Okay, so those would be 1, 2, and that's 3 and 4 different topic areas that would fit with this essay. By the way, this essay could well come up in some other form, I reckon, this year. It's unlikely that this one, and that's 2024, it's unlikely that this one is going to come up again, but I think something like the importance of nucleotides and molecules derived from nucleotides could potentially come up, and that opens the door to anything DNA related, but also anything ATP related, NAD and FAD and NADP related. Okay, also cyclic AMP, so there's lots of stuff that could be added onto this. Okay, right, so I'm going to have a go at writing one paragraph from this essay, emphasizing both the content and the importance. I'll come back in a second, and then you can read it. Okay, I got carried away, and I wrote 2 paragraphs, kind of because I was trying to do this whole section here, and then I kind of realized that actually you can kind of get 2 paragraphs out of it, so DNA structure and then DNA replication. So my top paragraph here, DNA structure, my second paragraph here, DNA replication, with the gap between the 2 paragraphs right there. Okay, so what I suggest you do is have a quick read of these, try and identify where you think I'm communicating my knowledge of AO1, content and use of keywords, and where I am kind of trying to sell the importance of this particular aspect. That's AO2, okay? Read the paragraphs. Where's AO1? Where's AO2? Pause to read. Okay, let's look at AO1. So, pretty much this is all AO1 up here. Polymer of nucleotides, sugar deoxyribose. I'm talking about the structure. So this is just AO1. I'm just describing the structure using a lot of keywords, purines, pyrimidines, the width of the DNA molecule. That's a little bit of importance actually there. That's kind of important there. So that's like AO2, just about. A to T. I could have added in the number of hydrogen bonds. That might have been good as well, but you don't want to add in everything you know because you'll run out of time. So I think I was trying to be selective. So encoding information required for protein synthesis, that's kind of important. I probably could have sold it a bit better, like really demonstrated why this is important. But I'm planning, in this essay that I'm writing, I would probably elaborate on this encoding a lot in my next paragraph on this protein synthesis down here. I probably hit it there. Then I really get into importance. The structure of DNA is important for several reasons. Information carrying bases in the center of the molecule protects them to some extent from chemical damage, makes them a more stable store of information, talking about rate of mutation, and then trying to link that. So the link it to cancer. So the way to kind of really sell the importance is almost to imagine a sequence of dominoes standing. This is me trying to draw dominoes here. What happens if you knock the first domino? How does that then cause a cascading chain reaction? What would be the consequence? So what would be the consequence of that? And what would be the consequence of that? So more mutation means more mutations in cells in multicellular organisms. More mutation in those cells means more chance of cancer. Maybe even I didn't say this, but maybe even multicellular organisms may never have evolved if RNA was the store of information, because they just, the more cells you get in a multicellular organism, more chances of cancer. Human beings got about 75 trillion cells, so if we had RNA as our genetic material, maybe we wouldn't exist. Let's go on to the next paragraph. Now I'm talking about DNA replication. Again, I don't want to say too much. I want to focus just on the importance. So I do talk a bit about replication. I talk about helicase, the DNA strands separating, hydrogen bonds, the molecules left exposed, free nucleotides. I say complementary base pairing, parent strands of DNA. I talk about DNA polymerase. I probably should have said joining up the phosphodiester backbone. I'm thinking I should have added that terminology in. Would have been worth some credit, I think. And then over here, this is kind of where I get into importance. Exactly the same information as the original parent strand. Semi-conservative replication is another key word. And then this is, again, importance. Is it allows both for the production of new cells in an organism via mitosis. And then I'm trying to get this kind of cascade effect. So we've got mitosis, and also allows for the inheritance of genes. This is more where I'm doing the domino thing. So if we couldn't have inheritance of genes, evolution and the biodiversity of life on Earth would be impossible. Possibly I could elaborate on this more. So I could say that evolution and speciation, maybe I could have said, relies on existing genetic code. So we have an existing genetic code. Mutations change and modify that genetic code, resulting in, during speciation, new species. So if we couldn't have that stable inheritance with a little bit of mutation, we wouldn't have all these different types of life. The key takeaway from this is that when you're doing these paragraphs, first thing, be selective with your information. I probably wrote too much in both of these, to be honest. I just get carried away because I'm not under time pressure. And the second thing is make sure to try and have an equal kind of weighting for AO1 content, that's my yellow highlights, and AO2 importance of, that's my blue highlights. Okay, I've tried to do that in each paragraph. And if you can do 4 paragraphs like this, then you should be on the sort of top marks. Okay, so that was the A-level biology paper 3. Lots of evaluate and a nice essay there at the end. I hope you found that useful, and best of luck in your upcoming exams. Bye-bye.